So today I'll be presenting the results of my PhD research, which I completed last year at the University of Cambridge. And as the title suggests, I'll be talking about lipid residues in pottery, specifically from Northwest India. So food is integral to any society. And in archaeology, to understand foodways or subsistence practices, we have to consider a variety of different factors and their relationship to one another. In archaeology, we are particularly good at interrogating questions about environment, and we have a lot of information quite often about the different resources that people have access to. But when it comes to actually understanding how food was processed or prepared, or to what extent it was considered edible or not, that is quite challenging and difficult to access. And including when we have to study foodways, which is the whole interrelated system of food conceptualization and preparation and distribution and consumption, we are better at understanding, arriving at questions of distribution rather than really, or consumption rather than preparation. But biomolecules in archaeology are, I think, providing us a way to actually have insight into these questions. And as you see, well, especially, I won't be talking about DNA or proteins or carbohydrates here, but a variety of these different biomolecules can actually give us access into the ingredients that are used and how they are prepared. These different biomolecules have variable preservation or contamination. They have degrees that affect them. And lipids, which I will be focusing on, they preserve very well relative to other biomolecules and are comparatively less affected by contamination, especially when you compare them to something like DNA. Contamination can be sort of identified quite readily in lipids, usually. Something else that's quite important to consider is that organic residues are not found in their natural environment. So there's quite little chance of their survival unless they're actually associated with something inorganic. So, which is why I'm looking at lipids in pottery. So just a little more information about lipid preservation and the different factors affecting how lipids are preserved in pottery. So lipids come through the foodstuff when food is being prepared in a vessel, and essentially they are absorbed into the ceramic. And you might have instances of a charred residue forming on the surface of the ceramic, but quite often you have what is simply, an absorbed residue, which is microscopic, you cannot see it, it's actually bound into the ceramic itself. And all sorts of different factors actually affect the degree to which lipids can be absorbed. So the type of the vessel, how it was made, its porosity, whether sealant was applied on it, how many times the vessel was used and for what products, secondary considerations, deposition, and of course the depositional conditions themselves will affect how lipids are preserved. We know, for example, that acidic soils actually are quite good at preserving lipids, but alkaline soils not so much. And finally, when it comes to actually excavating the pottery, you can introduce a lot of contaminants into the vessel at this time. So if you apply glue to fix the vessel, or if you store the vessel in a plastic bag, you could have, or if you wash the vessel or put vinegar in it, which is practiced in some regions, you are of course affecting the degree to which lipids will preserve or how much lipids are lost. So where do we find them? When we talk about lipids, we're talking about fats, oils, and waxes. And the reason they have a higher chance of survival is because they are hydrophobic. So even when you wash a vessel, you might get loose surface residues, but you still have a higher chance of them preserving. Something interesting about lipid residues is that experimental research has shown that it represents the total average use of a vessel. So not just the first or the last, but over time, lipids accumulate in the vessel from a variety of different uses. And some experimental research has shown that they tend to, if you are cooking fatty or boiling fatty or oily things, they will accumulate to the top, they'll float to the top. So you have a higher chance of acquiring lipids if you target the rims or the filling point of a vessel. But lipids also degrade and change. So this, for example, is a chromatogram of a fresh bovine adipose tissue, right? And you see these, if you just look at this in comparison, this is an early Bronze Age ceramic. So you have a complete loss of these complex molecules, and you're left with fatty acids. Fa stands for fatty acids. So you have a change in the distribution of fatty acids, and you have diagenetic alteration and a change in distribution. And these factors are quite important to consider when you're interpreting your data. So how do we actually acquire lipids from vessels? We drill or we crush 1 gram of this vessel, and we use different extraction techniques. Most studies will try to use solvent extraction because this preserves complex molecules, and you can, if they have even survived in the vessel in the first place. A more aggressive technique and yet very efficient technique is the acidified methanol extraction. And this is what I used in my PhD. And that's simply because this technique enables a very high output of lipids, and you can use this in scenarios where you do not have very good lipid preservation. Once you have your extract, you analyze it through gas chromatography mass spectrometry, where essentially you are separating mixtures of these organic compounds and you're identifying them on the basis of their mass to charge ratio of the ions. And what you might find then are lipids that are specific to plant or animal resources. So for example, something that's often detected in vessels is evidence for fish oils, but specifically those that have been heated. And you need to have a suite of different molecules to be absolutely sure that you have this product. Or you might have fatty acids, which are general to plant and animal sources. So very common molecules that you might find are the remnants of animal fats, the remnants of triglycerols, and these are fatty acids, so C16 and C18. And depending on your questions, you can access a variety of different questions in archaeology. You could talk about the technology and uses of vessels. You, of course, identify natural substances or foodstuffs, and you could even talk about the geographical origin of some of these products. Another technique that is applied is compound-specific isotopic analysis. And this is particularly applied to profiles or to extracts that look like animal fats, because this helps us distinguish between different animal fats. But there are fundamental differences in ruminant, such as ruminant animals and non-ruminant animals in their digestive physiology. And there are also differences in how fats are synthesized in the ruminant mammary gland and the adipose tissue. So we can use this information to distinguish between ruminant dairy, ruminant adipose, and then non-ruminant adipose fats. On the left, you see a very similar type of plot to what we normally use, but here is actually they've used local references, so local modern references from the region of animals, and compared their archaeological values to the local references. If you do not have local references from the region you're working in, you can use a sort of worldwide comparison, which is what you see on the right here. And this also helps you actually understand the input of diet into the animal's diet. So you can see the input of C3 versus C4 diet into the animal that was eventually consumed, or if the dairy was used, through this plot. So the values in green here and in red, these represent an increased C4 diet of the animal that was eventually processed in the vessel. So my PhD focused on analyzing lipid residues in pottery from a range of different vessels and from different settlements in Haryana, including very small villages and rural settlements, as well as the town of Farmana and Rakhigari, the city. And this was in collaboration with, I got, of course, access to samples from Farmana and Rakhigari through Deccan College. And the pottery I received from, I worked with Cameron Petrie and the Land, Water and Settlement and Terrains project to analyze vessels from the smaller settlements in Haryana. And we already know we have very interesting data and information about the varied cropping strategies that many of these smaller settlements were using. So we know, of course, that they're experiencing both winter and summer monsoonal rain, and they're growing a variety of different crops, and of course, summer and winter pulses and fruits as well. And quite importantly, we see a predominance of millets, which are C4 plants that are being grown. When it comes to animal exploitation, we have studies from the settlements that I was studying that, of course, quite similar to other regions in the Indus Civilization. There's a predominance of cattle and buffalo bones, a little less of sheep and goat, and the presence of domestic and wild pig. And of course, there is a very small proportion, but it exists, of wild animals such as deer, peafowl, riverine fish, hare, and even some mollusks. So there is already indications of quite a wide range of food resources available to Indus populations in this region. Something else, another aspect that's really interesting and that we know now is through enamel carbonate isotopic analyses. So this plot here, this work was done by Lightfoot et al., published recently, shows you that cattle and buffalo were consuming a high amount of C4 C4 plants throughout their lifetimes, compared to sheep and goat, which had a more varied diet. And we have only one example of a pig that seems to be eating C3 diet. So already here, this tells us a little bit about differential animal management, and that perhaps, as Kalyan actually presented yesterday, similar to in Gujarat, it seems that even in Haryana, there's cattle and buffalo are being fed C4 plants throughout, very consistently. And this might indicate some kind of unique foddering practice. And this information is important because it helps me understand, helps me interpret my lipid data later on. So what did I investigate in my PhD? Firstly, I was interested in understanding the degree of preservation of lipids in the vessels. That was an important methodological question, as not many studies have been conducted on lipid residues in the region at all. So what products were being processed in the vessels, and if there was a relationship between the form of the vessel and what was actually inside, what it was used for, and if there were differences and similarities in the contents of vessels, especially in the urban period, between different sites, and then over time. So I looked at both, as I mentioned, rural settlements and urban in the settlements. And I had access to vessels from the mature Harappan period as well as the late Harappan period. And I focused primarily on domestic pottery. And to supplement this information, I considered the evidence I already had from archaeobotany, from zoo archaeology, and from the enamel isotopic analyses. So what were the results? Essentially, 74% of the vessels did have lipid concentrations above 5 micrograms per gram, which is considered the interpretable threshold. But I have to clarify that I used a very aggressive technique, and there was a lot of loss of, complex molecules. So lipids were preserved, but only fatty acids were really well preserved in the vessels. But interestingly, when I compared pottery that had been washed and was lying in plastic bags for many years and had sort of varying degrees of contextual information associated with it. The lipid preservation was not so different to that of unwashed pottery, the pottery I collected myself from the field, and from which I had very good contextual information. So this might suggest that washing of the pottery doesn't actually, affect the lipid concentration as much, and the lipid preservation lipids had already degraded significantly, and it wasn't post-excavation that caused lipid loss. All the vessels I investigated had evidence of animal fats. So for example, ledge jars, which are often called cooking jars in the literature, had very characteristic fatty acid profiles of animal fats, which include medium chain and long chain fatty acids. These odd and branched chain fatty acids that you see here, which are supposed to be indicative of ruminant fats. And interestingly, they had relatively high lipid concentrations compared to other vessels. Perforated vessels as well. So these are very interesting vessels, as we all know, and people have been thinking about what they were used for several years now. And these profiles were also very indicative of animal fats. And so I was hypothesizing that perhaps these are then used for dairy production as cheese strainers, which previous studies have indicated. And other studies from Europe have also suggested that perforated vessels were used for dairy production or cheese production. But when I conducted the compound-specific isotopic analyses, I was very surprised by my results. I was expecting to find perhaps a lot more evidence for dairy. And as you see here, there are only 4 vessels that really show very clear direct evidence for dairy use in the vessels. And when I say dairy, I don't mean just milk. It could also mean other types of dairy products. A majority of the vessels seem to concentrate around in this region, which is indicative of the use of non-ruminant fats or those of ruminant adipose fats. And as we know, if we compare this to the faunal record, we know that there's actually very limited evidence of non-ruminants. So pigs, for example, are only about 2 to 3% NISP. So, that's interesting and intriguing. And similarly, we have a predominance of cattle and buffalo bones, but actually not as much ruminant adipose fats as I would expect. And one thing that we can actually pinpoint and be fairly clear about is because we know that cattle and buffalo were quite consistently feeding on C4 plants, and this plot can help us isolate animals that had a very high C4 diet, we can be quite sure that the dairy products were coming from cattle or buffalo, and that some of these values are probably quite clearly indicative of cattle or buffalo meat processing. These values are a bit more harder to explain. They could be sheep and goat, they could be wild deer. But these indeed are still a challenge to explain. And I'll give you some more hypotheses or explanations later in the presentation. But let's compare what we see, let's compare the different settlements. So for example, from Farmana and Rakhigari, we see a very similar spread of values. Rakhigari had extremely large ranges in the isotopic values, but this is not really as easy to explain. It could perhaps mean that animals from the region or from Rakhigari had access to very variable diets. Perhaps we're talking about animals coming from different parts that are eventually ending up in the pots and being consumed in Rakhigari. But from Farmana, we have some really nice complementary data that we can use with the lipid residues. And that is of starch grain analysis, which is conducted by Arunima Kashyap and Steve Weber. So they found evidence of horse gram and curcuma turmeric from cooking pots in Fermanagh. And while I'm still waiting for the full published results, it's a really interesting way to complement the lipid residues, because we actually can see evidence of plant processing in the vessels. Whereas with lipids, it's a little more difficult to access plants because they simply, animal fats just are fattier, they have more lipid in them, essentially. When we look at the small settlements, we see again a very similar spread of value. So there's no difference between the urban settlements and the rural settlements. And a very similar pattern, very limited, it's still there, but there's very limited evidence for dairy processing, and a similar concentration of these values in this non-ruminant region. And as we know, there's a very diverse assemblage of archaeobotanical remains from a number of these sites. And we know that millets are grown more frequently and in greater proportions than wheat or barley. And yet we have most of these values concentrating with showing evidence of more C3 input in whatever they were cooking. So how do I explain this? I will get to that, but essentially some other questions that I asked were if there was a relationship between the vessel form and the vessel contents. And when I look at simply lipid concentrations, it's not so easy to say, oh yes, these vessels were used more often or they had very high lipid concentrations. Some vessels showed relatively higher lipid concentrations, like ledge jars, as I mentioned earlier. But I analyzed very few bowls, for example. I focused on more jars that were used for cooking. So it's not, I would have to analyze probably a lot more vessels to really arrive at any solid conclusions about this. But there's slight indications of ledge jars being used, perhaps for cooking fatty products. And finally, the perforated vessels did not show any evidence for dairy processing. And this was really intriguing and confusing result, because most of them, again, plot in this non-ruminant fat region, except for a single one from Fermanagh, actually. And if I compare this with the results from Kotada Bhadil that Kalyan presented just yesterday, we see that he has a lot more evidence for dairy compared to what I have, and several of the vessels that he analyzed were bowls. The dairy vessels were bowls. So it's possible that I did not analyze the right type of vessels. Perhaps bowls were used more for dairy processing, but there seem to be differences. We are maybe talking about regional differences in vessel usage and in cooking preferences here. So it would be really interesting to explore these questions a bit more and to do a proper regional, a comparison of different regions. Now here's where I explain a little bit about the complications of interpreting this data. So we know that resolving mixtures of plant and animal products can be quite challenging in lipid isotopic studies. So some hypothetical mixing experiments have been conducted, essentially, where you'll notice that as you increase the contribution of plant in a vessel, so this is an example of dairy product with a plant, you have a higher chance of achieving values that fall within non-ruminant fats as you increase your plant contribution. And similarly, if you mix a plant with a ruminant adipose fat, if you increase slightly your plant contribution, you again might have values that resemble non-ruminant fats. And there are further difficulties with applying this method when it comes to environments which have both C3 and C4 plants. So if you look here, if you have a C3 plant with a dairy product that is coming from an animal that ate mostly C4 plants, you have a chance of getting values that look like meat. And if you mix a C4 plant with an animal that was fed entirely on C3 plants, you might have values that falsely look like dairy products. So these mixtures could actually confuse your results, and you need to be quite careful when you think about, you need to consider these essentially when you're interpreting your data. So what was being processed in the vessels? I have a lot of these very high positive big delta values. Are they actually non-ruminants, or are they mixtures of dairy products and C3 plants, or of ruminant fats and plants? It could be both. It is very possible, it would be actually really interesting if non-ruminants were preferably cooked in vessels, but it is a bit challenging to explain this as the faunal record simply doesn't show such a high predominance of non-ruminants. On the other hand, it could be that cattle and buffalo or meat was preferably charred and cooked or roasted and consumed in this way. So we could be talking about two different modes of consumption. One is boiling and the other is roasting. So to conclude, the technique really opens up new ways of looking at pottery, moving beyond actually looking at the cultural role that pottery played. I mentioned earlier that there might be regional variations across the Indus civilization, and it would be really interesting to explore these questions. And thirdly, the prevalence of dairy products is another really interesting question to explore in different settlements. And is it really missing in the settlements I'm looking at, or was it mixed with other products? And one of the most important things we need at this moment is local modern fats and oils, so that we can do a much more better comparison to our archaeological data. And I'm currently trying to collect these modern fats. It's a bit challenging because I need animals, I need the meat of animals and I know that they should have had a controlled diet. I need to know exactly what they ate in their lifetime. So it's a bit complicated to actually to procure these fats, but I'm working on it. And something else that I think would really benefit, especially in this region, is a bit more experimental research in the field. Trying different mixtures of fats and animals, of animal fats and plants, and seeing what you get.